Today, we are talking about a totally revolutionary type of cancer therapy, one that acts less like a medicine and more like a precision-guided weapon. They're called Antibody-Drug Conjugates, or ADCs for short, and they are completely changing how we fight one of our oldest enemies.
For more than 100 years, this has been the single biggest challenge in cancer treatment. How do you kill the cancer cells, which are really just our own cells that have gone haywire, without destroying all the healthy tissue right next to them? It's a problem that has defined the fight against cancer for decades.
For a long time, our best weapon was traditional chemotherapy. And you can really think of that as carpet bombing. I mean, it's pretty effective at killing cells that divide quickly, which cancer cells definitely do. The problem? So do your hair follicles, the lining of your stomach, your immune cells. The collateral damage can be just brutal. But what if you didn't have to carpet bomb? What if you could launch a precision strike instead? Well, that is the entire idea behind ADCs, a therapy that is designed from the ground up to seek and destroy only the cancer cells, leaving everything else alone.
But this idea of a precision strike, it's not actually new. All the way back in 1910, this visionary scientist named Paul Ehrlich dreamed of creating what he called magic bullets. These would be compounds that could just hunt down a specific disease in the body and wipe it out without causing any other harm. For over a century, that was just a dream. But today, that dream is finally becoming a reality.
So how did we finally build this magic bullet? Well, we basically had to create a biological missile. And just like any advanced piece of military hardware, it has three absolutely critical components that have to work together perfectly.
First things first, you need a guidance system. In this case, that's the antibody. It's a protein that's been engineered to be a kind of master detective. It's searching for one specific clue, a protein called an antigen that shows up all over the surface of a cancer cell, but is pretty rare on healthy cells. When it finds its target, it latches on and it does not let go.
Okay, next up, every missile needs a warhead. For an ADC, this is the payload, an incredibly potent cell-killing drug. And I mean, we're talking about poisons that are thousands of times stronger than anything you'd find in traditional chemotherapy. They are way too toxic to just inject into someone by themselves.
So why does the payload have to be so ridiculously powerful? This number says it all. After you inject an ADC, only about 2% of the total dose actually makes it to the tumor. Just 2%. With such a tiny fraction arriving at the destination, each and every one of those missiles has to pack an absolutely enormous punch to get the job done.
So you have your guidance system and you have your warhead. Connecting them is the third and maybe the cleverest part of all, the linker. You can think of it as the detonation trigger. It's a chemical bridge. And its job is so important. It has to be rock solid and stable while the missile is traveling through the bloodstream, keeping that poison locked up. But it's also designed to be smart, to break apart and release the payload only when the ADC is safely inside the target cancer cell.
All right, so now we know all the parts of our biological missile. Let's watch it in action. Its mission to take out a cancer cell is a really precise four-step operation.
It all kicks off with targeting. The ADC zips through the bloodstream until its antibody finds that one specific cancer cell and locks on. Then comes infiltration. The cancer cell doesn't know what's happening and actually pulls the whole ADC inside. That's when we get to detonation. Once it's inside, the cell's own internal environment, its chemistry, breaks that smart linker, unleashing the toxic payload. And finally, elimination. That freed poison gets to work, completely destroying the cell from the inside out.
But there's another amazing feature here, kind of like a secret weapon. Some of these ADCs are designed so that once the payload is released, it can actually seep out of that first cancer cell and go kill its neighbors. This is called the bystander effect, and it is a massive advantage. It means you can destroy cancer cells in a tumor even if they don't have that specific target protein on their surface. It just amplifies the whole attack.
Now, as you can probably imagine, getting all three of these pieces, the antibody, the payload, the linker, to work together in perfect harmony, yeah, that took a ton of trial and error. This technology has been evolving for decades, getting smarter and way more effective with each new generation.
The journey really began with Ehrlich's idea back in 1910. Then, a huge leap forward in 1975 gave us the ability to make those monoclonal antibodies, the guidance systems. It took until the year 2000 for the very first ADC to get approved, but it proved the concept could work. By 2013, the second-generation drugs were showing massive improvements. And since 2019, we've seen this explosion of third-generation ADCs that are more precise and predictable than we ever thought possible. So what exactly got better over time? Well, everything. They refined every single component. The first generation, that was the proof of concept, but it used antibodies that weren't very human-like and had unstable linkers that sometimes dropped the payload too early. The second generation fixed a lot of that, with better antibodies and much more stable linkers. And the third generation? Oh, now we're talking fully humanized antibodies, super stable linkers that have a controlled release, and brand new, extremely potent payloads. The result is a much more predictable and effective drug.
And the results of all this hard work, they really speak for themselves. After that first approval in 2000, progress was kind of slow and steady. But in the last few years, the field has just exploded. By the end of 2021, the number of approved ADCs had jumped to 14. 14 different drugs, fighting all kinds of solid tumors and blood cancers.
So this isn't just theory anymore. These drugs are out there on the front lines, making a real impact on patients' lives. Let's just look at a few quick examples, and then peek at what the future might hold for this incredible technology.
I mean, drugs like Kadcyla and HER2 have completely transformed how we treat HER2-positive breast cancer. Adcetris has had a massive impact for patients with certain lymphomas, and Padcev gave us a powerful new weapon against advanced bladder cancer. These are just a few examples of how ADCs are becoming a real cornerstone of modern cancer therapy.
And the innovation is not slowing down. The next wave is even more sophisticated. Just imagine missiles with 2 guidance systems for even better accuracy, or warheads that carry 2 different kinds of poison to defeat drug resistance. Scientists are even developing smaller ADCs, more like darts that can get deeper into solid tumors. And some are even swapping out the toxic payload entirely for one that basically calls in the patient's own immune system to join the fight.
All of this leads to one final huge question. As these biological missiles get smarter, more powerful, and more precise with every single new generation, are we finally getting close to the day when the carpet bombing of traditional chemotherapy becomes nothing more than a relic of medical history?